Starting. All right. Yeah. So hello everyone. My name is Shubham and welcome to our second Discrete Diffusion reading group, which I'm co-organizing with Justin and Jihan. Now I'll quickly go over the logistics before getting started with the talk. We meet on Mondays every day at 10 a.m. Eastern and 4 p.m. Central. All the talks are recorded and uploaded on our YouTube channel. We have uploaded the talk for the previous session on our YouTube page. A link to it can be found on our website. And also, please add this calendar invite that was shared with everybody who had subscribed to the Google group, so that way you don't miss any sessions. And also, if there are any changes, it's easier for you to stay on top of those things. So we announce all these talks on email and Twitter. So follow us on Twitter so that you can stay, so that you don't miss out on any of these talk announcements. We also have a Discord channel. I highly recommend joining Discord because not everybody gets a chance to ask questions in the reading group. So we have channels such as paper discussions where we have threads specific to the papers that are presented in this reading group. And also there are other channels like self-promotion. So for instance, if you're a PhD student looking to get into this area and you want to share some of the cool projects you've worked on, this is a really good good platform for doing that. And also if you have openings on your team, be it in academia or industry, this is a channel where you would announce jobs specific to discrete diffusion. And all the job announcements, PhD positions or full-time positions, intern positions, they're made on Discord and also will be shared on our website. Okay, so in today's talk, we'll shift focus from language generation to a different modality, molecule generation. Unlike language, which follows a left-to-right inductive bias, molecules have no such structure, making them particularly well-suited for discrete diffusion. We'll discuss a paper from Professor Pranam's lab called Peptune, which was led by Sophia, who is an undergrad at UPenn and not Duke. Pranam's group has produced several impactful contributions in this area, and this work continues that strong line of research. I believe Pranam's lab is hiring PhD students in this space, so I'll let him quickly introduce his team and the research that they're working on. And so Pranam, feel free to take over.
Hi, Shubham. Thank you so much. Thanks everyone for showing up to Sophia's talk. I'm just going to give a very short intro, and let Sophia handle most of today's meeting. So my name is Pranam. As Shubham mentioned, we're at Penn in both bioengineering and computer science. Our lab is really interesting, where half the lab are mostly pure AI scientists developing new generative models. The other half are experimentalists who actually take what comes out of those models and translates them not only in the lab, but also in animals and eventually, hopefully, to the clinic. So this, discrete diffusion is very close to my heart and close to the heart of a lot of my students. We came across it about, maybe a year and a half ago, actually at Subham's talk at ICML, at a random ICML workshop, where, in biology, we've been looking for something that does two things, right? Is generative, can go from, completely masked sequence to a fully unmasked sequence or a fully generated sequence, and one that is bidirectional, because we struggle in biology knowing that proteins interact and molecules interact in both left to right and right to left and all these other weird directions. And so, when we saw MDLM and we saw the discrete diffusion space growing, we were like, well, this could be the solution that we've been looking for, at least to generate molecules. And our first example was with membrane proteins, and we actually validated in the lab. But when Sophia joined, Sophia came to me and really was excited about doing molecule design with AI. Sophia's background largely was experimental with some computational background, but I told her, hey, the ultimate question is what she's showing you right here, which is saying, can we design therapeutics but make sure those therapeutics have all the properties that you want? But Sophia just joined, so I told her to try something simpler, just generate peptides. But just a few days later, Sophia gave me or maybe a week later, she gave me an overleaf where she
Pranam, I think you're muted.
Can you hear me now?
Yes.
Where did you last hear me?
Last 30 seconds, maybe.
Okay, cool.
So you were telling, Sophia, shared this overleaf with you, and after that we lost everything.
Yeah, exactly. Sorry. Yeah, so Sophia shared this overleaf with me. I think this is almost our first lab meeting where she was just an undergrad, and she shared it, and it pretty much mapped out, hey, here's discrete diffusion, but here's how one way you could do multi-objective guidance, which is what she's going to present to you today. I will tell you just as what has happened since Peptune, so Sophia will fill in the rest. Peptune we've utilized to generate therapeutic peptides that we've been synthesizing in the lab for various different disease targets that Sophia will talk about. But Peptune has also been very applicable to many other projects in our lab. For example, targeting maybe specific disease proteins, but not their wild type counterparts. Sophie, another graduate student in the lab, is working on that. We've used it for mRNA design. So discrete diffusion overall, and Peptune specifically, has been the backbone largely for all the new algorithms and therapeutics that we've developed in the lab that are now in cells and are moving into animals. So if you need any motivation on why I think discrete diffusion and also discrete flow matching and other variants are so exciting, this talk should really motivate you. So with that, I want to welcome and I want to introduce Sophia. She's an undergrad in my lab. This is her first paper in the lab, and she's already produced 5 this year since joining. So hopefully not only will you enjoy this talk, but definitely take a look at all of her other works. She has more work coming out. She's really a superstar, and I'm very fortunate and lucky that she chose to work with me. So Sophia, take it away.
Great. Thank you for this wonderful introduction. So obviously today I will be talking about Peptune, my first paper in Pranam's lab, and it's a paper on how we can generate peptides, therapeutic peptides with multi-objective guided discrete diffusion. Okay, so a brief overview of the agenda that I have planned. I'll just go over a brief primer of discrete diffusion, which I'm sure many of you are familiar with, and how we were able to inject controllability in the unmasking time of discrete diffusion. And then I'll go over our multi-objective guidance algorithm for discrete diffusion, and then a bit about what's next and kind of a hint on like the newer work that we have done in this space, and a conclusion and obviously Q&A. Feel free to kind of stop me whenever you have a question. I'm happy to answer throughout the presentation as well. Okay, so a brief overview of mass discrete diffusion. We basically have this clean sequence X0, and we want to inject noise, and this is done with a forward discrete diffusion process. And we basically inject noise as masked tokens, and these are kind of like pseudo tokens that represent any kind of distribution of tokens that can be generated. And in the unmasking process or the backward denoising process, we want to be able to recover this clean sequence from a sequence of masks. And during the training process, what we do is we basically have this input sequence, and we inject noise with a probability denoted alpha. So this is the probability that it remains unmasked at time t. And then you can think of this as drawing from a categorical distribution where alpha is multiplying the clean sequence and 1 minus alpha is multiplying a just complete sequence of masks. And then we inject this into a bidirectional transformer. And this transformer model basically predicts a distribution over each of the masked tokens. So you can see like denoted X alpha, X theta is a kind of distribution over the token space that is predicted by this transformer model. And we basically want to minimize the difference between the predicted distribution of tokens with the ground truth distribution. So a ground truth token is just a one-hot vector. And you can see that once these two kind of distributions match, the loss becomes zero. So we really want to minimize kind of the difference that the model is predicting with the true sequence.
Oh, Sophia, one clarification question. Can we? Oh, so essentially you're saying, on the left-hand side, you have your clean sequence at the bottom. You're showing how to go from a clean sequence to a mass sequence. And that mass sequence is being fed into this like transformer with bidirectional attention. And the model is, so you're asking the model to reconstruct the missing tokens, right? Like that's what the cross entropy loss is at the bottom. Okay, cool. And there's a weighting term, I believe, like in AR model, I believe you just have the cross entropy loss. That is, like the cross entropy between the predicted token and the true token. But in diffusion models, you have additionally you have this weighting term 1 over lowercase t, right?
Yeah, so this is determined by basically the masking schedule that you use to mask the tokens, and it recovers kind of like a loss that's weighted by how far along the unmasking process is. So when you're closer to a fully unmasked sequence, you want to like weight the loss higher, and that's kind of what this term controls.
Okay, so you're saying if your mass sequence has fewer mass tokens, then the loss should be weighted lower, and that if it has weighted higher, and if it has more mass tokens, it should be weighted lower, and that. Okay, cool. Thank you.
Yes. And then for the inference process, we basically start with a fully masked sequence, and we want to recover some sequence from the data set. So we have this, like, suppose we have already unmasked a few tokens, and we have this partially masked sequence. And then we feed it into the same transformer model, but this time it's now trained. So it already has learned kind of how to generate this token sequences from the data distribution. And then we sample from the categorical distribution, similarly to before when we injected the noise, but this time we're kind of reverse, we're sampling from a reverse distribution where this clean sequence is now replaced with this parameterized model, the prediction from the model. And then this gives a cleaner sequence. We basically unmasked a few tokens, and eventually we will recover some clean sequence that hopefully represents a sequence from the data distribution.
Oh, one follow-up. Sorry, I'm being very annoying here. But okay, let's try to like understand that categorical distribution that you have. So you're saying, okay, you have a partially masked sequence that, the sequence with mass tokens, you feed that into the bidirectional transformer, and that your transformer gives you probability distribution for the mass tokens, right? So I believe that categorical distribution that you have, you're sort of like flipping a coin and deciding, hey, should I unmask a token or not? And if you choose to unmask it, you draw a token from that X theta distribution modeled by the transformer. Is that correct, or?
Yes.
Okay, cool, great. Yeah.
And why we decided to use discrete diffusion specifically for our task, which was biological design, or more specifically peptide design. First of all, biological sequences naturally contain these long-range bidirectional dependencies. So while with like language, you have these kind of sequential dependencies that are usually the case where you have like the prefix is kind of most of the context needed to predict the next token. With biological sequences, that's not ever really the case, and you always have these bidirectional dependencies. And as I just mentioned, diffusion avoids this positional bias inherent in autoregressive models, where you have this prefix that is kind of determining the next token that is generated, and we don't usually want that in biological design. And discrete diffusion also leaves room for controllability in the unmasking order, which we will discuss a bit more later. But in autoregressive models, you can only unmask the next token, and you can't really control like which position to unmask or to reveal next. And diffusion kind of gives you a bit more flexibility on that end. Okay, so I'll also go over a bit of motivation behind our specific work, Peptune. And the question we really wanted to solve was, can we generate valid therapeutic peptides that are simultaneously optimized across multiple properties? So peptides are these kind of drug-like sequence, kind of like small molecules, but longer, and kind of like proteins, but shorter than proteins. And they have these drug-like properties that can really be optimized through generative processes. And this is kind of like the model that we wanted to create. And there really lacked a good multi-objective guidance strategy for discrete diffusion. As I'll discuss a bit more later, there is kind of a lot of limitations with classical guidance techniques on the discrete state space. And we really wanted to be able to create a good algorithm that can do multi-objective guidance. And there's also a lack of generative models for non-natural and cyclic peptides. So peptides, there are 20 like natural amino acids that you see often in proteins, but peptides, they can be modified to have better therapeutic properties. And there wasn't a generative model that could accurately kind of generate sequences that had these non-natural amino acids and these non-natural modifications to enhance these therapeutic properties that we want. And first, I'll kind of go over why random unmasking order fails to preserve these key sequence structure properties of peptides, but also any type of sequence that has like some sort of structural component that needs to be unmasked first. So with random order unmasking, usually like a mass discrete diffusion model, it doesn't have really bias on any position, and it can unmask in any order depending on just like the categorical distribution I explained earlier. And it kind of gives this random context, and you're predicting the distribution over the mass tokens with this random context. And then you could, in the best case scenario, you'll get a good sequence, but in most cases, this can cause kind of invalid sequence structure where this random context was not sufficient in predicting the rest of the sequence. And if we have this like structural components where these parts of the sequence are very necessary for predicting the rest of the sequence, we can unmask those first. And then we have this context that is now kind of like better suited for predicting better context for predicting the clean sequence. And in this case, it's easier to get a valid sequence from the distribution. And in the case that we wanted to explore was peptides. So peptides, they have these bonds between amino acids that kind of are necessary for both in reality, when you want to synthesize a peptide, you have these amino acids that you need to connect through the peptide bond. And when these peptide bonds are not present, it becomes an invalid peptide. So we basically really want to get this context in first, and we want to be able to unmask these tokens first. So the big question was, how can we enforce this early unmasking of this necessary context during inference? So in the case of peptides, that would be peptide bonds.
One quick question. No, I think this is a really good motivation for discrete diffusion as well. So if someone were to do this using an autoregressive model, because there are guidance techniques for autoregressive model as well, but I think the biggest limitation there would be that because you'll be doing left to right generation, there's no way for you to generate the peptide bonds first and then generate the remaining molecules, right? Am I correct, or?
Yes. So peptide bonds can occur anywhere across the
Yes.
sequence. So with autoregressive models, you have to generate sequentially, and you wouldn't be able to enforce kind of where to generate first. So if you wanted to generate the peptide bonds, that wouldn't be possible with autoregressive models. And so to do this, we kind of introduced this bond-dependent masking schedule, and we really wanted to encourage accurate unmasking of peptide bond tokens prior to the rest of the sequence. And so here you can kind of see the same process, the same forward masking process as I shown before, but this time, instead of alpha being the same across the entire sequence, we now have this alternative alpha where it's 1 minus t raised to the power of w. And this weight kind of enforces that we have later masking of peptide bond tokens. So this power basically decreases the value of t. T is between 0 and 1, so it becomes a smaller value. And then we have that the probability of it remaining the ground truth token is larger, and so it becomes late masking of those tokens. And we show in the next few slides that this actually enforces early unmasking of these peptide bond tokens during the reverse process. And so you can kind of also see like the categorical distribution. This time, alpha is no longer the same kind of value for the entire sequence. It's now modified for those peptide bond tokens.
I'm going to jump in quickly and just wanted to quickly mention that this is very specific to how biology works, right? In biology, peptides are connected because, as Sophia mentioned, they're connected by these bonds. If we just masked the tokens like equivalently, then what could happen is in the reverse process, there'll be a lot of examples where the peptide will be fine, but the bonds are messed up. And if the bonds are messed up, then it's not even a peptide, really. So I think the goal here was to reduce the masking rate, or at least enforce like late masking of peptide bonds so that we are ensuring that those are going to be generated correctly, right? And then the model is able to worry about filling in I would say, the important but not as critical regions of the peptide sequence later. It's a very unique thing, probably almost specifically for biological sequences.
Pranav, do you mind if I ask a quick question here?
Yeah.
So yeah, I'm curious. So once you generate these peptide bonds, like you have, let's say let's say 7 like mass tokens in between. What does that like number 7 like mean? Does that mean like how many amino acids that you can fit into the two? Yeah.
Yeah, I mean, Sophia, you can answer. I'll just do a quick thing. So usually, because what Sophia's doing here is she's not representing these as amino acids anymore. She's representing these as chemical, almost like chemical side groups, if you can imagine. These are groups of atoms that are basically telling us what this peptide is and what it does. And so in between the bond tokens are the composition of atoms which are represented and tokenized that represent a specific amino acid. And so now instead of saying one letter, right, that represents an amino acid, now you have a collection of tokens that together can represent the atom. This gives us very fine-grained control over what the amino acid can be. And it actually allows us to explore chemistries that may not just be the 20, existing ones. This so that if Sophia wants to, as you'll see later, wants to get these really specific and optimized peptides, we are able to like find the exact amino acid with the exact atoms that give us those properties. So it's actually much more nuanced than say like what a protein model would do, in which they wouldn't even have bond tokens. They would just put the amino acids side by side together. So does that make sense? Those 7 in the middle, in your case, are just atoms that represent an amino acid, in rather than one token that does so.
Gotcha.
Yeah.
Thanks.
Okay, so this we showed that in the paper that this alternative masking schedule actually induces this very special bond-dependent loss. So we have this, when we derive the Novo loss from the forward masking schedule, we now have this weighted cross-entropy term. So instead of just the kind of log probability, I realize now that it's a bit denoted differently from previously, but basically we have this log, this cross entropy term, but now instead of 1/t, we have W/t. So it's a larger weight on the loss term. And so it gives you this kind of loss that is half the peptide bond tokens are now weighted with this extra W that increases its loss, and the rest of the tokens are now weighted with just the regular negative cross entropy term.
Just wanted to say, does that make sense for everyone? Because it's probably I think what was one of the coolest innovations that Sophia and Novo came up with, because in this case, like if the peptide sequence is invalid, right? Meaning that and the only way it's invalid realistically is if the peptide bond is not there, because then we can't actually make it in the lab. So it's almost like an experimentally like kind of enforced loss, right? Because if Sophia gave us a sequence and those peptide bonds were, random token or wasn't the right token, then the experimentalist in the lab just would be like, I can't do anything with it. So it's important that we're upweighting that loss, while the other stuff, if it's not a great token at the other amino acid positions, Johanna, as you were mentioning, then we'll deal. But in this case, we have to get the peptide bonds correct. And the fact that we are getting that lost term in there helps us out of that in that case.
Yeah, and in the paper we also describe how we have this kind of decoder that can convert the SMILE sequences to a peptide if it is a valid peptide, but if it's invalid and these peptide bonds are out of place, it will kind of just return invalid and it's just not a peptide. And so this also introduces this very specific reverse posterior. So before you could kind of see that the categorical distribution was a weighted sum of the clean sequence and a mask sequence. And so when we have this different kind of schedule in the forward process, we have this also different reverse categorical distribution. And you can see now that with the peptide bonds, you have this extra W that's like the kind of probability of it being unmasked to the true token is now raised to the power of W, and that actually decreases its value. So this is a value between 0 and 1. And so when you raise that to a power, it becomes smaller. And so the probability of becoming the clean token in the reverse process at earlier unmasking steps, so like closer to 1, is larger. So you can see that peptide bonds have a larger probability of being unmasked at earlier time steps in the reverse process and smaller probability of remaining a mask. And you kind of see this kind of like complementary style where you have this late masking during the forward process, which corresponds to early unmasking in the reverse process. And we show with this training, after training with this alternative schedule on a bunch of different peptide SMILEs, we show that we get really high validity actually, and a lot of non-natural amino acids, which was very exciting because a lot of models have these representations where you can't really you can't represent this larger space of amino acids where there's a bunch of different modifications that are so diverse that you wouldn't be able to tokenize them each, or the probability would just be so small. But with SMILES tokenization, it's very special where they're so granular that you can generate any type of molecule and any type of peptide. And we were able to generate peptides that both had these modifications, but also were valid, so they can be synthesized in the lab. And they can also be cyclic, so they can have these modifications where two ends of the peptide can actually connect and form this cyclic peptide that has enhanced therapeutic properties. And now I'll go a bit more.
Quick question. So during generation, right? So you have this waiting term W for the peptide bonds. So yeah, so so how does the generation work? So you would not know like which positions to assign. If you can quickly walk me through the generation process. I'm a bit confused as like how you pick the weighting. If you don't know a given mass position is going to lead to a peptide bond. Maybe that part was not very clear to me.
Yeah, so we actually, in the reverse process, we let we show that when we derive the reverse posterior, it is like it's weighted higher for peptide bond tokens, but since in the generation process, so it's just like kind of like the theoretical idea that it will be like the model will just naturally predict those peptide bonds first.
Oh, okay. So some weighting term will, so the probabilities assigned to the peptide bonds will be weighted according to the weight W in some way, I guess. Like those will be upweighted. Yeah. So that, okay, cool. No, cool. Makes sense.
Yeah, and during the forward process, we actually have this this decoder that's able to detect where the peptide bonds are. So that's how we actually induce the weights in the forward process. Okay, and now I'll just go over kind of our multi-objective guidance process, and I'll first introduce guidance for discrete diffusion. So for example, we have this unconditional model that's generating a bunch of peptides from this data distribution. And this data distribution has like every peptide that you can ever think of. And it doesn't have to be a good therapeutic, it could just be a random peptide. But what if we wanted to optimize certain properties where instead of the data distribution, we have this distribution with property Y? So for example, these peptides all bind to a specific target protein of interest, and that's kind of the property Y. And this is an alternative distribution that's kind of different from the data distribution, but also on the same space of molecules. And so what if we wanted to optimize multiple objectives? So for single objective, there are methods to do this, which I'll discuss also later. But for our case, we wanted to optimize multiple objectives. And so first, I'll want to discuss, define kind of what multi-objective guidance even is. So you can kind of think of single objective guidance, very easy to know what the maximum value of the reward is. But with multiple objectives, you kind of get this like trade-off scenario where there are multiple different optimal sequences that can be optimized in different areas or different rewards in the kind of set of rewards that you want to optimize. And so we can define a term called Pareto dominance. So when a sequence is Pareto dominant to another sequence, it is no worse than the other sequences in the kind of set of Pareto optimal sequences in any objective. So all the scores are either greater or equivalent to all the rest of the sequences in the space. And it also has to be better in strictly one objective, or at least one objective, strictly better in a certain property. And we can also define Pareto non-dominance, where we basically have this sequence that is not dominated by any other sequence in the space of all possible sequences or in the Pareto optimal set in this case.
Sorry, I have a quick follow-up question about the very first equation, like for Pareto dominance. So it says for all k, skx star is greater than or equal to skx, okay? And there exists a k prime such that, what is sk prime?
So that's the score in the k prime
Actually, it's one objective. There's no, x star is no worse than x in any objective. I see.
Yeah.
Cool.
Yeah, so S denotes kind of the scalar reward in one objective, and K kind of it's the index of the objective. And so with this non-dominant kind of constraint, we can get this Pareto optimal set. So every sequence in the set is not dominated by any other sequence in the set. So you kind of get this like Pareto front where you have these sequences that are optimized across all objectives and maximize the trade-offs that you can possibly get from the space of all sequences. So now I'll kind of discuss the challenges of guidance in the discrete state space. So in continuous diffusion, we have this two kind of classes of guidance strategies. We have classifier-based guidance where we have this alternative, like this added term to this unconditional logits. So you can think of the last term as the unconditional logits, and this is the probability of the specific property given this noisy sequence. And for classifier-free guidance, we have the same thing, but now we can train a model that's also conditioned on a given property. And this also, both of these strategies kind of introduce this extra term that is not compatible exactly in the discrete state space, and I'll explain why. So basically, there are several limitations with this. First of all, with multi-objectives, we have like scalar scores, but we have multiple of them. So we don't just have one scalar score that we can inject in this gradient term. So how do we scalarize is a question. And usually we don't want to scalarize at all because scalarizing means that we're kind of reducing all, like we're losing all the kind of trade-off information that we have between the objectives, and not necessarily having an optimized scalarized score means that you have like a Pareto optimal sequence. And it also doesn't balance conflicting objectives, as I just mentioned. So when you scalarize, you don't really know which objective was better, and you just get this one score that doesn't really represent the multiple objectives that you want to optimize. And this also in the discrete like state space, you can kind of see that the ZT and the ZS terms, these are both noisy sequences. So they're not clean sequences. They are kind of partially masked in the case of discrete diffusion. So you would have to evaluate scores somehow on these like partially masked sequences, which is not always the easiest thing and can give very noisy reward values as well. And this also only produces a single sequence for each run through the model. So if you want to do guidance and generate one sequence, that's fine. But with multi-objectives, you can kind of see before that you have the set of optimized sequences. And just generating one sequence independently from all the rest is not the best way to go about generating like a Pareto optimal set of sequences. Okay, so now I'll kind of introduce our algorithm that overcomes these limitations and is directly applicable to mass discrete diffusion and is able to kind of optimize multiple objectives without scalarizing. So the first step in this process, we basically have 4 steps, and this is based off the common Monte Carlo tree search algorithm, and we apply this to discrete diffusion. So the first step is to select a unmasking path with high potential reward. So suppose you already have this tree of many possible unmasking steps, and we basically want to find a path that has the highest potential reward. So the highest potential to generate a sequence that has high rewards when evaluated with these predictor or models that predict the reward of a certain sequence. And so
Sorry, quick question.
Yes.
Sorry, when you say select a trajectory that has highest potential reward, do you draw multiple samples? So for instance, you have ZT, then you'll probably sample 20 different candidates for ZT minus 1, and then you pick the best one. Like, do you do greedy? You sample a bunch of candidates and then you pick the best best one and then move on to the next denoising step, or how does it work?
Yeah, so we kind of, since this is like an iterative process, we kind of start here with like, suppose you have this like tree of existing like unmasking steps, and we want to select kind of among these like which one is the best step to take that are already like we've already explored these, and which one is the most optimal in like the next step. So I'll discuss later kind of like the expansion of the sequence, like to explore multiple different unmasking steps, but in this case we just want to like traverse a optimal trajectory to some terminal node that hasn't been expanded yet or hasn't been explored.
Okay. Oh, sorry. My question was, how do you even tell like which sequence is better than the other? Like, do you feed this partially masked sequences to a separate classifier, or there's some other way?
Yeah, so that's in the next step. We
Okay.
have like a rollout step, and then we evaluate rewards and then back propagate it.
Okay.
kind
Sorry.
of it's a bit confusing because it's an iterative process. So you can't really
Okay.
start anywhere and be able to have the full context. So
Okay.
I'll go over the entire algorithm and hopefully it makes
Cool.
sense by the end.
Sure. Sounds good. Yeah.
Okay, so you have this selection reward. So you can kind of see this as just like the reward across multiple objectives, and this is a vector, so it's not scalarized. We have like the raw rewards of each objective separately in a vector form, and we normalize this by the number of visits to the nodes. So we don't want to like overvisit a node and have that be like overvisited more and more just because it's visited so many times. It has a higher reward, and this kind of extra term encourages exploration of unvisited child nodes. So if a node hasn't been visited at all, this term would be higher, and that would encourage a bit more exploration instead of just going to the high reward unmasking steps. And then this kind of determines a trajectory to a terminal node. And then as someone was saying, like we want to kind of explore different unmasking steps. So in this step, we basically expand a terminal node. So you can think of this as like a partially unmasked sequence. And we explore multiple different kind of unmasking strategies. So like one single unmasking step, but multiple different ways of unmasking the sequence. So you can kind of think of this as like we have this like distribution predicted by the model, and we apply some noise, so some stochastic noise that can perturb the distribution a little bit, such that we don't get kind of the same distribution at all the different unmasking or expansion steps. So this is trying to kind of explore like multiple different ways we can unmask the sequence. And then the next step, to obtain a clean sequence, as we mentioned before, we don't want to evaluate rewards on noisy sequences because that's very hard, especially in the biological sense. You can't really feed a masked sequence into a reward model and get a good quality reward. So we want to roll out each sequence such that it becomes a clean sequence. And we do this with greedy unmasking. So we just sample greedily from this categorical distribution predicted by the model, and we get this clean sequence, which we feed then into different classifier models. So in the case of peptides, we want to optimize, for example, target protein binding affinity, cell membrane permeability, therapeutic properties like non-toxicity. And so we feed this into these black box models. You can kind of think of them as black boxes for now, that returns a reward or a scalar reward for each sequence. So it takes this clean sequence, we feed in the model and gives a reward, and then we have this vector of scores. And now we want to kind of return, like we
Sorry,
want to.
one very silly question about the vector of scores. So it's a vector, so it assigns a score to every token in a given sequence. Is that so? What is the length of the vector? Is it the length of the partially masked sequence, or?
Yeah, so the score vector is the length of the score vector is how many objectives there are. So there's only
How many
one
objectives?
vector for each sequence.
Yes. One vector for each sequence. Cool.
Yeah, I wanted to quickly say that I think the beauty of this is that we're scoring the sequences after they're generated, right? So we're never like, so and Sophia, correct me if I'm saying anything incorrectly, but the sequence is already fully unmasked at that point. We score it. This is also good because the classifiers that we have in the lab that Sophia will mention, like cell permeability or target binding affinity, or like you can pretty much give it as many objectives as you want. Sometimes they may be black boxed, right? So we don't even know, we cannot train in like a noisy predictor of those sequences. And so this actually allows you to say, oh, random person in the other side of the world has trained a black box predictor of solubility. We can take it, you can still score it, right? Your fully unmasked sequence, and then you can use that, as Sophia mentioned, to backprop the rewards based off its score, right? And its comparison to other sequences that you've rolled out. So yeah, it makes it a lot more flexible.
Mm-hmm. Yeah, and then for the backpropagation step, as Pranan mentioned a bit, we have these like child sequences that we've expanded, and now we've rolled out each of these kind of nodes into a fully unmasked sequence, evaluated the multiple objectives on each of them, and then we have multiple reward vectors now, so one corresponding to each of these nodes, and we basically sum them up, and we get this reward vector based on how optimal these sequence compares against the existing set of Pareto optimal sequences that we've already generated. So you can kind of see P star as a set of sequences that have already been generated from the model. And we compare the newly generated sequences with those set of sequences. And we get this kind of vector that's now showing how optimal the sequence is across each of the objectives against this set of sequences that we've already generated. And then we add these child rewards to all the predecessor nodes that were used to generate these expansion steps. So all these nodes kind of like are unmasking steps that gradually kind of produce these child nodes. And we want to up reward them so that in the next iteration, we visit them again if they have high rewards, and we don't visit them if they have low rewards or they didn't produce any good sequences. And then in this process, we kind of iterate multiple times. And so each iteration, we start again at this fully masked sequence. We go down and choose a good trajectory. We expand to get like these child nodes. We evaluate these on different rewards, and then we backpropagate. But then we see that at each iteration, we actually generate M different sequences, and they're already evaluated on all these properties. And so we add the Pareto optimal sequences to the set that we kind of have going across this whole process. And this is what kind of determines what sequences get added in the next step. And at the end of this whole iteration process, we generate a total of n iterations times m child sequences at each iteration. And so we have this Pareto optimal set across all of those generations. And those are kind of a representation of the most optimal sequences across multiple objectives. And this, as Pranam mentioned in the beginning of this talk, this can be applied to multiple different discrete diffusion frameworks, not just peptides. It can apply to mRNA, small molecules, DNA, RNA, and even maybe to language if we want to do a specific reasoning task and we want to optimize, for example, correctness in multiple aspects. This can also be applied to that. And the good thing about this is it can scale to any set of properties without additional training. So you don't have to train the model at all. And we just have to have good classifiers or good verification methods for predicting these rewards from clean sequences. And it also balances exploration of diverse unmasking paths and exploitation of high reward paths that you can see during the iteration process. And it handles conflicting properties by maintaining the set of Pareto non-dominated sequences. So during this whole process, you don't really discard any sequences, and you always kind of keep these good sequences that you've already generated. And so at the end, you get this really optimal set of sequences. And it can, as we will describe later, it can optimize binding affinity with multiple protein target sequences, but in any scenario, it can kind of optimize all these rewards, and these rewards don't have to have gradients, they don't have to be some sort of neural network. They can be black box reward models, and this algorithm can be able to maximize all these rewards. And so in our case, we applied this to peptides specifically. And so we showed that our model is able to optimize binding affinity to very popular targets. So this is GLP-1, and we know that it's a very popular target in the past. And we showed that against these commercially available peptide drugs, our peptides generated have lower docking scores, which indicates stronger binding affinity. So it is generating very optimized peptides for binding affinity. And we also show here in the next slide that you can see kind of visually the iteration of this guidance process, and you can see the scores gradually being optimized across the iterations. And this is multiple different objectives. So this is 5 different objectives that are all therapeutic properties, and they're all being simultaneously optimized across the iterations. And this is just another validation. We do docking. So docking is kind of a way that we, in the lab, kind of in silico validate whether a peptide is binding to a protein structurally and physically. And so we see that we have very optimized docking scores. Anything below -6 is very good in terms of docking. And we're able to kind of get these peptide binders that have very low and very high binding affinity to the target.
Oh, one thing I wanted to mention, Sophia, if you go back to that, just so that you guys get an understanding of why this is useful, when you want to put these peptides into a mouse or, a human, I think there's a lot of considerations that we have to think about. Solubility, hemolysis is basically toxicity because it kills the red blood cells. Non-fouling is are you sticky to random things, which you shouldn't be. So I think 2 things. The limitation, I would say, of this whole strategy is that we need good predictors. And those are largely dependent on the supervised trained models, right, the score functions that were pre-trained. And that, is data that you would have to collect ahead of time. But once you have those predictors and you trust them, then, as Sophia mentioned, it's super easy to just plug them in because you don't need to retrain the base generative model, and you can just score for the reward vector generation, you can just score across any of these predictors. So, and the beauty is that you could use them in different ways. And what Sophia showed on the left is something I don't want you guys to not consider, is that you can even use the same predictor twice, right, for different contexts. For binding affinity, we had both transferrin and GLAST, and we wanted to bind to both. And so we could even incorporate that. So it just shows you, hopefully, the flexibility of the model, and that you could take any base generative model, whether it's peptides, proteins, mRNA, whatever, and any set of predictors for that modality, and you could put them together. I think it's pretty flexible, and it's pretty cool.
Yeah. And then for conclusions, why why is this model significant? Obviously, we can modulate these, like we showed that we can modulate masking schedules for different tokens, and this can be useful in preserving these fundamental structures in the data beyond just peptide bonds. And any sort of sequence that has these fundamental structures, we can apply this kind of method to upweight the unmasking process for those key structural components. And this guidance strategy can kind of provide a generalized framework for multi-objective guidance for discrete diffusion models, and it doesn't require any additional training and can scale to any set of objectives. And I just want to kind of hint at some newer work that we've done. So we have this guidance strategy, so we can kind of apply it during inference and have multiple objectives optimized, but the kind of cost of searching through or iterating through the tree search process is a bit expensive. And what if we want to kind of train the model so that it can unconditionally generate these multi-objective optimized sequences? And this can be thought of as like amortizing the search cost by improving the underlying model weights. And we can, a strategy for doing this is off-policy RL, which is basically a fine-tuning strategy that enables learning from diffusion trajectories from a replay buffer. And we can reweight these sequences generated such that they kind of are optimized so they have the same distribution as a reward tilted distribution. So it's no longer a data distribution, it's actually a reward tilted distribution that becomes the generation target of the unconditional model. And to do this, we can fine tune using off-policy RL and using diffusion trajectories optimized with the same algorithm that we've described earlier. But now we are changing the model weight so that it's always going to generate these highly optimal sequences. And this is just a newer kind of hint at some newer work that we've done with some collaborators where we have this tree search algorithm, but now we're modifying the model weights in a way that makes sure it's theoretically guaranteed to converge to generating from a optimal reward tilted distribution. And I won't go over too much in detail of this paper, but if you're curious, feel free to read the paper. And this is kind of more recent work since Peptune. It's actually been out for quite a long time. So I just wanted to highlight some more recent work that we've done. And we actually showed that this new algorithm that we have, this fine-tuning method, can generate peptides that are even better than Peptune optimized peptides, just because we are fundamentally changing the weights so that it's always generating better and better peptides, even as the search goes on. We're modifying the model weights that in the next iteration, the unconditional distribution that you're optimizing, that you're guiding for, is actually even more optimized than before. And we show that this can even better optimize across multiple objectives.
Quick question. So do you train that algorithm from scratch using RL, or you take a pre-trained model and then you apply some RL fine-tuning on top of it for it to generate?
We take a pre-trained model and then we apply RL.
Oh, pretty interesting, yeah.
Yeah, so that's all for me. I am open to any questions that you have, and yeah, feel free to read the Peptune paper, and then this is the new paper that I just mentioned.
Yeah, thank you, Sophia, for the great talk. I think we should take questions now. I see in the chat a lot of the questions were answered by Pranam. So yeah, I'll.
Sorry, Sophia.
No, this is great.
I didn't see any of the questions, so that's great because I can't see anything right now. I can just see my screen, so that's great. Thank you.
Yeah, so folks, feel free to ask questions now if you have any. Okay, we have Justin.
Yes, so thanks a lot, Sophia. The talk was very interesting. And yeah, I had one question for the rollout part. So my understanding is that now when you want to generate a peptide, you start from full mask, you do the full denoising, and then you check whether the path is good or not based on the full sampling process. So I think so this is I assume is quite costly. And I was thinking if you had tried something like, for example, you start from a certain node, let's say noise level 0.3, like T equals 0.3. And the posterior for mask diffusion gives you the possibility to sample directly from X0 in one step. So it will not be as good as if you do the full sampling like in multiple steps. But I was curious if sampling from this in one step and judging computing the different scores can give you something informative about the rollout from this point compared to doing the actual rollout. Is it also something, because I assume then it could speed up the process even though it's not correct. So I was curious if you try something like that. Yeah.
Yeah, that's a great suggestion. I think for us, since peptides have these like very intricate structures where you can't really evaluate the reward of an invalid peptide. So if we unmask all the tokens that are masked at the same time, we get this, like, it's very likely that we get a very invalid peptide because there's no dependencies whatsoever when you're unmasking. And although it's faster, we wouldn't be able to evaluate correctly the properties of this peptide because the models, like the reward models that we have, they're kind of trained on valid peptides. So even though that this can actually like is a great idea for when there isn't like such intricate structures in the molecule. So if you have a protein that's just 20 amino acids, like I think that's a great speed up that can have very informative like rewards on like the intermediate state of the sequence. But in our case, since we're doing peptides that have these structures that need kind of the extra decoding steps to get a valid peptide, we didn't do that. But that's a great suggestion.
I see. Yeah, that makes sense. Yeah, thanks for the answer.
Now we have another Justin who wants to ask a question. So Justin number 2.
Hi, super great talk. So thank you for presenting. I wanted to make sure if it'd be possible to clarify like the training and then generation. So for training, the denoiser takes in the positional encoding of where the tokens are in the sequence, the time step embedding, but then it also takes in like this kind of embedding of whether a token is the bond or not. Is that correct?
Not exactly. So in the training process, we like it's the bond only determines how when it's being masked. So the probability of it remaining masked or remaining the ground truth token, that's what the peptide bond kind of determines. We don't actually like there is like probably in the embedding itself, there is some information about whether it's a peptide bond or not, but we don't actually inject that information like specifically, oh, it's a peptide bond, it's not a peptide bond inside the model. So that's just determining like the masking schedule during training, not the actual input to the model.
Okay, got it. That makes sense. Right. All right, thank you.
Sorry, next we have Tejumai.
Yeah, hi. Thank you for the talk. So just maybe a naive question. I wanted to ask whether like the tree search algorithm that you implemented, is it like equivalent or a modified version of the A-star search algorithm, something like that?
I don't think I'm familiar with the A-star search algorithm, but it is like a modification of the Monte Carlo tree search algorithm, which has been used for like reasoning tasks for yeah, I'm not sure I'm familiar with the A-star search.
Oh, okay, sorry for the question then.
It's okay. My fault for not knowing.
No
It's okay.
worries.
All right. Yeah. So do we have any other questions or we wrap it up? Yes. There's a question from Mert.
Hello. Thanks for the talk again. So as far as I remember from Mr. in biology, peptides are these like somewhat one-dimensional chains of proteins, but when amino acids, but when you combine them into proteins, they can acquire interesting geometry. Is that at all part of your generation process, or could that be incorporated to make it more efficient or maybe easier to?
Yeah, that's a great question. So, while proteins do have these very like defined three-dimensional structures, for peptides, they're much shorter. So they have like around like 10 is the average amount of amino acids in a peptide, and that can form some structures, but they're not defined three-dimensional structures. And this kind of like using like a structural representation of the peptide. Usually there isn't like a defined structural representation. It's very flexible and it can kind of change over time as it binds to a protein, as it evolves. So we chose to use sequence representations, which we do a lot in the lab. If you follow us ever, we do a lot of sequence representations, but the reason is because we think that a lot of the information about the peptide, a lot of the structural and also like property information is contained within the embeddings generated from the sequence. So we have these rich embeddings, which is a high dimensional space, kind of like the structural space, but more informative in some sense because it's not defining a static structure. And that is kind of informing the properties and then the prediction process of the peptide. But yeah, that's a great question. I think for proteins, it's very valuable to have like this three-dimensional structure. But in the case of peptides where it's smaller, more flexible, can adopt many different conformations at any different times, we chose to use a sequence representation.
Yeah, thanks. That makes sense. Appreciate it.
Yes. Yeah. Any other questions or?
I'm answering just the last few in the chat, so if you hold on to closing the thing.
Yeah. I mean, yeah. No. Thanks for doing that. But yeah, I mean, if you have any other questions, like we can take it on Discord. Like there are specific channels. There's one for this particular, like it's called paper discussions. We have created a thread just for this particular paper. So if you have any questions, feel free to post it there. And yeah, thank you so much, Sophia. Thank you so much, Pranam. It was a wonderful talk. And yeah, thank you for your time, everybody. So yeah.
Yeah, thank you for
Bye.
joining.
Of course. All right. Bye.
Thank you, guys. See ya.
See ya.